Hello, I'm Mrs. Covarrubias, and I'm going to be doing your cell test review with you tonight. So this is Chapter 3 in your book. Make sure that you use your online book. There's review games and interactive things that you can do. And then after we're done with this, make sure you study this information and look over all the different note-taking guides and things that we did in class. Okay, so the first thing we're going to talk about is the cell theory. This is important because learning about the cell, we need to know where it came from and kind of the rules of what makes cells cells. So the first one is that all organisms are made of cells. All organisms are made of cells. Okay, so you cannot be a living thing and not be made out of a cell. You can have one cell, and that's okay. It's a single-celled organism, but if you, like an organelle, for instance, is not a living thing because it's not made of cells. It makes up cells. The next one is that existing cells are made cells come from existing cells. So cells come from existing cells, and this is really alluding to a chapter we're going to do later, that is mitosis, how cells reproduce, and they reproduce by splitting. Hopefully you remember talking about that in middle school. The last one is that cell is the most basic unit of life. So cells are the basic unit of life. All right, the 5 scientists that were really important. Our first one was Hooke, and he was the one that first saw cells and named cells. Named and saw first cell. And then there was many people after him within the next 100 and 200 years that did a lot of work on the cell. The next one was Leeuwenhoek. And he was able to see him in greater detail. He looked at like his teeth scrapings. He named them the animalcules. Animalcules. So you see these little tiny creatures moving around and that no one had ever seen before. And those are the bacteria that live in our mouth and our teeth. Okay, Schleiden was the next one, and he was known for finding plant cells. And then we had Schwann. And he did animal cells. And then our last one was Virchow, and he kind of settled the ongoing debate of the last bit of the cell theory, like where are these cells coming from? Are they just popping up? Do they just come from nowhere? And he is the one that understood this process of mitosis, or this that cells come from existing cells. And that's how we grow and reproduce. All right, the next thing that we need to talk about are prokaryotic and eukaryotic cells. So we were talking a little bit about Leeuwenhoek's animalcules. Those were prokaryotes, and those are bacteria. And bacteria, they are very small. They're about the size of a mitochondria inside of our cells. So they're much smaller, about the 1/10 of the size, I think is what the book says. They have cytoplasm, so that's something that is the same. They also have a cell membrane. But they're very old. They're a lot older than what our cells are. They were around Earth before we were. They also have no nucleus. However, they do have DNA. They have a region of DNA, but they don't have a membrane-bound nucleus that's housing their DNA. And they are very, very simple. They do not have complex organelles like the mitochondria and the Golgi apparatus producing vesicles carrying proteins, and the rough ER and the smooth ER. And so we say that our cells are very complex organelles, and these are very simple. They do have ribosomes, though. They do produce proteins. And this is what makes us sick, the proteins that they produce. And so a lot of the antibiotics, they go in and they stop the function of the ribosomes, or they disrupt the cell membrane. That's how antibiotics work. So eukaryotic cells, on the other hand, they do have a nucleus. They're very complex. They're very much larger. They're also very high functioning in comparison. So we're able to put out a lot more proteins and carry out a lot more functions than our simpler bacteria. However, bacteria can sure make us sick by their simple function. And so eukaryotics, this is us, this is our cells, and this can either be us, like animal cells, or these can be plant cells. So we have plant and animal cells. Also, the fungi and protists fall into that category, but we didn't really touch on those. Okay, and here's our two types of eukaryotic cells. So we have plant cells and animal cells, and we want to know the differences and similarities of animal cells and plant cells, and they have some extra organelles that animal cells do not have. So the organelles only in plant cells are going to be our chloroplast, and these chloroplasts are going to be turning the sun into food. And we don't have those, unfortunately. We can't lay out under the sun and gather enough energy to make food. We have to eat food, but plants can't do that. They also have the central vacuole. And we have vacuoles, but we don't have as big as vacuoles. And this is water storage for the plant. And these are very important because a plant needs lots of water to make food. Water is actually broken down when they make their food from the sun. And so if they don't have water, this process can't happen. And so these two organelles kind of work hand in hand. The next one is the cell wall. Okay, we don't have cell walls, and it serves as extra structure and support for plants. And that's why they can sprout up through, the dirt, and they really are, quite rigid. And we're a little softer. Our bones give us that structure. And so if we were just cells, we'd be pretty mushy. But plant cells, they're really strong because of their cell wall. Organelles that are only in animals, this is like our lysosomes. There's some plants that have some lysosomes, but most of the time lysosomes are considered to be only in animal cells. And that's pretty much the only thing that a animal cell has that plant cells don't, because plant cells have mitochondria. Even though they have chloroplasts to make ATP, they use mitochondria in their cells as well. So they have more organelles than what we do. All right, so let's talk about these organelles. We have the cell membrane, and this is going to be a semi-permeable membrane, and it's really going to gauge what goes in and out of the cell, and it also serves as structure for the cell. Okay, remember this is made out of phospholipids. We're going to talk more about this in the question below. All right, the nucleus, this is storage for genetic information. Okay, so this is where all of our DNA is. This is where RNA is made, and these are the instructions for the cell. Okay, and they give instructions to the rest of the cell, and then that's going to make proteins that give us, all the things that we're able to do and how we look. The nucleolus is that center region inside of the nucleus that produces ribosomes. Vacuoles are going to be like little closets that store all sorts of things. They can store vitamins for us. They can store water. They can store other nutrients that we might need later. Okay. Vesicles, these are little membrane-bound transport containers. Okay, and what these are going to do, these are produced by the Golgi apparatus. So we have the Golgi apparatus that has all these layers, and these are spitting out these little vesicles, and they hold proteins, and they're going to ship things around the cells, and they're kind of like these just little transport containers that move stuff from one side of the cell to the other, or they move out to the cell membrane where they kind of latch on and exit proteins or exit other materials through the process of exocytosis. Okay, mitochondria, these are the energy producers. These are going to take glucose and they are going to break it down into ATP. And we're going to learn all about that this coming chapter or standard. Ribosomes, these are the protein producers. They are producing proteins that are going to travel all over our body and do all sorts of different functions for us. Rough endoplasmic reticulum, these hold ribosomes and aid in protein production. Smooth endoplasmic reticulum, this is actually going to modify the protein. So after it gets done at the rough ER, the protein is going to move over to the smooth ER and it's going to be modified. Especially enzymes, they have that really specific active site for the lock and key model. And so this is what's making sure that they're folded in the correct position. And the smooth ER also detoxifies the things that we eat and the things that we drink go into our cells, and the smooth endoplasmic reticulum is what takes that toxin in, and it changes into a molecule that's not toxic and disposes of it for us. All right, lysosomes, these carry enzymes. And lysosomes are kind of hanging out around the cell, and they're waiting for something that organelle that is not functioning correctly, or food that needs to be digested. The lysosomes go in, they engulf it, and they break it down with the enzymes that they have inside of their capsule. And this is really important because enzymes can't just be free-flowing, or they would be breaking down all of the functioning organelles. So lysosomes are really important because they're the capsules that carry the enzymes. Okay, the Golgi body or the Golgi apparatus, this is the shipment. of proteins. They're going to make those little vesicles that I was talking about here, and they're going to ship the proteins out of the cell. Cytoskeleton, these are the microtubules and the microfilaments that allow organelles to move throughout the cell, and they also serve as structure for the cell. They almost are like little interstates all through the inside of the cell, holding it, giving it its shape, and allowing things to move within the cell with little motor proteins. All right, centrioles, these are those, they almost look like little pieces of licorice or something. We're going to draw them below. But they are going to aid in cell division. When we're talking about the cell reproducing and the cells coming from existing cells, these centrioles are responsible for that. So they move to opposite sides of the cell, they pull on the microtubules, microfilaments, and they actually split the cell into two during cell division. And cytoplasm is the jelly-like substance that holds organelles, kind of is the inside of the cell. All right, our next question is talking about drawing the cell. So we're going to first draw an animal cell. Okay, so we're going to draw the cell membrane here. We'd have the nucleus with the nucleolus inside. Okay, we're going to have the rough-ER that surrounds it out here, and in order to make it the rough-ER, we would want to put little dots all over it. You know that it is rough-ER if there's tons of dots. Typically our smooth-ER is out here. Okay, not going to have any dots. Might be smaller than that, depends on the type of cell. Our liver cells are going to have lots of smooth ER because that's where a lot of our blood is filtered and a lot of the toxins go. So we have our smooth ER, and then your Golgi apparatus is going to be like little sacks stacked on top of each other. No dots on those either, no ribosomes. And the ribosomes will be throughout the cytoplasm. And then we're going to have our mitochondria that has the folds in it like this. We would have our lysosomes that have some kind of funky structure. I always draw mine like this, like little, I don't know, starfish or something. We're going to have some vacuoles here that are storing material for us. Okay. Lysosomes and vacuoles, and we're going to also have some vesicles that are transporting things around the cell that are produced from this Golgi apparatus, so there'd be lots of vesicles around our Golgi. And some of them were connecting to the outside, probably letting proteins out. And then we would also have, let's see, what am I missing here? Let's go back up and look. Nucleolus, vacuoles, vesicles, mitochondria, ribosomes, smooth ER, cytoskeleton, centrioles. All right, cytoskeleton and centrioles. So my cytoskeleton is going to be kind of like all these, like, if I could draw littler lines, it would be kind of like these little tiny lines throughout, all through here, around the outside, and really, truly, they go all throughout the cell, but typically we don't draw that because it would just look too messy. And then what else? Oh, centrioles. Yeah, so the centrioles kind of hide out, and they are these little structures that kind of look like this. I picture them to be really heavy and dense because they're going to be real strong and pull everything apart. So they're like these things that kind of stack, and then when it's time for the cell to divide, one will stay on this side and the other one will go clear to the other side. Okay, so that's our animal cell. Our plant cell is going to be a little more rigid. It's going to have this cell wall here, and then on the inside of that, we're still going to have our cell membrane made out of phospholipids. Okay, we'll still have our nucleus, and we'll have a nucleolus in there. Probably won't have any of our lysosomes, but we're going to have mitochondria. Not as many mitochondria as an animal cell typically. Then we have our chloroplast, and these have little circles in them. That's how you know that they're chloroplast. These are what we're later going to find out to be called grana or granum inside there. So those are chloroplasts. Then we're going to have a real large vacuole. We're not going to have centrioles. Oops, I should have put that on. Let's go fix that real quick. Over here. There's no centrioles in a animal, in a plant cell, because a plant cell divides a little differently. So up there, go ahead and put that. We're still going to have our roughy R here with our ribosomes. We're going to have our smoothie R. Still going to have a Golgi apparatus. We're still going to have vesicles around, produced by the Golgi apparatus. We're going to have ribosomes all over. Plants produce proteins just like we do. Okay. And let's see, what else am I missing here? Golgi apparatus, large vesicle. They're also going to have cytoskeleton on the outside, giving them some more structure, and all the way throughout. I think I got it all. Ribosomes. Let's check up there again. I don't want to miss anything. Nucleolus, vacuole. We have the central vacuole as well. Vesicles, mitochondria, ribosomes. Smooth and roughy R. Lysosomes not in the plant cell. Golgi, cytoskeleton. No centrioles. Cytoplasm. Okay. And then we have large vacuole cell wall chloroplast. All right, we're good. So animal cell, plant cell, a lot similar, few differences. If you're able to draw it with me here, you should be able to label it on your test. All right, next one, describe the structure of the cell membrane. So the cell membrane is going to have phospholipids that encase most of it. They're going to have some proteins in them, some of them for structure, some of them for signaling. Okay, so we might have a protein here for signaling. And then you're going to have some for transport. Okay, and they're going to have a what we call transport channel. And they can either facilitate active or passive transport, depending on if they need energy or not. Our structural proteins aren't going to have anything on top of them. They're just going to be proteins by themselves that kind of add a little stability to the cell wall, or the cell membrane, excuse me. And then our signal or receptor ones are going to have these carbohydrate chains that stick up, and these are how the cell communicates. Okay, so these are our carbohydrate chain receptors. Oops, I forgot to draw my fatty acids on my glycerol here. Okay, so we have our phospholipids with their two fatty acid tails attached to the glycerol head here. Remember, these are molecules of carbon, and so the cell membrane is just so small and thin. I mean, even if we looked at these cells on a really high magnified microscope, we're not going to be able to see a lot of this structure as detailed as we're drawing it here. But we know from chemistry that these molecules look like this, so we're able to draw it. So inside there, we're also going to have some cholesterol. Okay, so remember, lipids are hydrophobic, and so they're really going to keep that water out, and this cholesterol is also going to add to the structure of that cell membrane to make it a little stronger and act as a very nice barrier for us. We're also going to have the cytoskeleton here inside of the cell that's attached to the cell membrane, and so it's going to come across and it's going to attach to these phospholipids and to the proteins, and it's going to serve as structure. Some of them will go out and across the cell like we were talking, through the cytoplasm, And then these little motor proteins actually walk on these microfilaments and they pull organelles across the cell. So they're like little highways inside the cell. Okay? And then these were our passive transport. We wouldn't have anything. If there was active transport, there would be a little spot here to get ATP. But these are the phospholipids, the cholesterol, so those are the lipids. The proteins, we have all three types. We have a structural protein, we have a signal protein, and we have a transport protein. Our carbohydrate is our signal or our receptor. Our cholesterol is in there, and our cytoskeleton. So there is our cell membrane. All right, next one. Talking about passive transport, there's three types. We talked about diffusion in class, and we talked about facilitated diffusion. Oops, I don't know what I'm spelling there. Hold on. There we go. Facilitated diffusion. And then we have osmosis. So these are our three types of passive transport, and they require no energy. Okay, so diffusion can be a gas or a liquid, but this is just something that's going to be able to cross the cell membrane with that concentration gradient. So that concentration gradient is when there's a high concentration on one side, so there's lots of oxygen molecules on one side of the membrane, and then you have your cell membrane, and then over here there's not. And so molecules are always going to go to the side that there's not as many molecules, and water is always going to want to go to the side that there's more molecules. And so in this case, oxygen is going to be able to pass freely through the cell membrane, which is great for us. Because oxygen and CO2 are great examples of diffusion because they don't need a protein to get past the cell membrane. They're able just to go across, so when we're running, oxygen can freely go into the cell and carbon dioxide can freely leave. Facilitated diffusion needs a protein. Needs a protein, but it does not need energy. These are also very specific, and so this is like insulin. Has a protein that it goes out of, and insulin is the only thing that can go out of the cell, and no other things can go out of that protein channel. And when you eat a whole bunch of sugar, then the insulin is able to go out of there. No energy needed, which is great because glucose is what we need to produce energy. So if we needed energy to let insulin out, that would cause a very big problem for us. And then osmosis is water movement. And we're going to talk about this in the question below here. Water movement across the cell membrane. And it also is going with that concentration gradient. And so the example of osmosis, it would be like when we're dehydrated. We eat too much salt, and then our cells lose water trying to make the concentrations equal. Or we drink too much water, and the cells start absorbing water and start rupturing, maybe, because there's too much water on the outside. So if the cell remains unchanged, we call this isotonic. Okay, and so these are the different solutions that cause osmosis. So isotonic causes no osmosis. The cell does not change, and the solution means that it's the same on both sides. When the cell swells, this is going to be a hypotonic solution. Tonic just means solution. Hypo, I always think of hippo. Hippos are big, and so hypo is going to make your cell swell, okay, or it's going to cause it to expand. And so this would be when you have something that's a really high concentration on the inside of the cell, or just higher. It doesn't have to be super high. and then you drink a whole bunch of water, so the concentration on the outside seems lower, okay? So then water is going to start moving in, because the whole purpose of osmosis is to make the concentrations the same. The concentrations have to be the same. Okay, so water moves in, it causes the cell to get bigger and bigger and bigger until maybe it pops. Okay, and then the last type of solution is when the cell shrinks. Okay, and this is going to be a hypertonic solution. And hyper, I think if people are really hyper and they're moving a lot, they're usually really small and skinny. And so this is going to cause the cell to shrink. And this is due to a concentration on the outside. So you go to McDonald's and you eat French fries, and they're really salty. And so you have a lot of salt on the outside of the cell. And so water starts moving out of the cell, and this causes the cell to shrink down and get shriveled and cause dehydration. So both of these are not good, but because we have osmosis, we're able to regulate that, and it helps our body. I mean, we don't have to think about it. It just happens naturally, and it keeps our body within range, and hopefully we choose healthy things to eat and not too much water because we're in a contest or something that make our cells do this often. All right, our last question talks about active transport, and the big thing here is you need energy. And this energy is ATP, which we're going to learn about next. So ATP allows these following active transports to work. So we have pumps, protein pumps. They're channels that kind of move things in and out. We have endocytosis, and we have exocytosis. I should write that out, I suppose. Okay, so protein pumps are going to move molecules against the concentration gradient. So there could be a whole bunch of sodium on the outside, and a protein pump will pump more sodium to the outside, or a hydrogen pump will pump more hydrogen into that area instead of letting it flow and make the concentration gradient the same. So these molecules move against the concentration gradient. An example of this is sodium and potassium pumps. And these are in our brain, and these are in our muscles, and they work with our nerves, and this is how action potential happens and how we are able to think and move. So sodium and potassium pumps. And if you were to look on YouTube right now and do active transport sodium potassium pumps, you'd be able to see them moving and see them moving these things against the concentration gradient. Okay, endocytosis is when you're going to see this word engulfs. Okay, when the cell engulfs something. We call this phagocytosis when a cell's eating, or when a white blood cell eats a bacteria, or when a cell takes in water. Okay, so engulfs something from the outside. And this is really the cell membrane. Cell membrane engulfs material. Okay, so this is like cell eating, or white blood cells killing bacteria from your immune system. Okay, exocytosis is when the cell membrane puts out material. And so those are those little vesicles that we were talking about, letting out proteins, or letting out water, or and the vesicle actually attaches to the cell membrane and then becomes part of the cell membrane to let it out. And so this would be proteins leaving the cell. All right, I hope this helped. Now study this information, know it for tomorrow, play some games on your online book, And do well on your test tomorrow. All right. Thanks for watching.